Greetings to everyone. The topic of our video today pertains to the captivating science of why our waistlines widen in middle age, driven by the surprising role of stem cells called adipocyte progenitor cells. Groundbreaking research from City of Hope, published in Science, reveals how aging activates these cells to produce new fat cells, particularly around the belly, fueling risks for chronic diseases like diabetes and heart conditions. By identifying a new type of stem cell, committed preadipocytes, age-specific, and a critical signaling pathway, scientists are paving the way for therapies to combat age-related obesity and enhance longevity. Without further delay, let us delve into our discussion. The City of Hope, a titan among cancer and diabetes research institutions in the United States, has illuminated the cellular mischief behind expanding waistlines in middle age. Their findings, unveiled in Science today, pinpoint a novel target for therapies aimed at curbing belly fat and prolonging vitality. Why do our midsections swell as years advance? The answer lies in adipocyte progenitor cells, a cadre of stem cells nestled within white adipose tissue, the culprit behind age-related heft. Aging, it seems, awakens these adipocyte progenitor cells with fierce vigor. Unlike most adult stem cells, whose vigor fades over time, adipocyte progenitor cells in middle-aged mice surge into action, spawning new fat cells with relentless zeal. While most adult stem cells' capacity to grow wanes with age, the opposite holds true with adipocyte progenitor cells. Aging unlocks these cells' power to evolve and spread, declared Adolfo Garcia Ocana, PhD, the Ruth B. and Robert K. Landman Endowed Chair in Gene Regulation and Drug Discovery Research at City of Hope. This marks the first evidence that our bellies balloon due to adipocyte progenitor cells' prolific output. Through meticulous mouse experiments, later mirrored in human cells, researchers unearthed a startling truth. Transplanting adipocyte progenitor cells from older mice into younger ones sparked a torrent of new fat cells, as if the older cells carried an intrinsic blueprint for expansion. Yet, when young adipocyte progenitor cells were placed in older mice, they remained sluggish, producing scant fat cells. Aging clearly equips adipocyte progenitor cells to churn out fat independently of their host's years. The research also unveiled a new breed of stem cell committed preadipocytes, age-specific. Emerging in middle age, these cells, propelled by a signaling pathway called leukemia inhibitory factor receptor, LIFR, vigorously generate fat cells, explaining the burgeoning waistlines of older mice. We discovered that the body's fat-making process is driven by LIFR. While young mice don't require this signal to make fat, older mice do, noted Chong Annabelle Wang, PhD, co-corresponding author and associate professor at City of Hope's Arthur Riggs Diabetes and Metabolism Research Institute. Human tissues, too, tell a parallel tale. Single-cell RNA sequencing revealed CPA cells in middle-aged individuals, brimming with the same capacity to spawn fat cells. This discovery underscores a universal mechanism across species. Our findings highlight the importance of controlling new fat cell formation to address age-related obesity, Wang emphasized, hinting at future medical breakthroughs to trim belly fat and enhance longevity. Belly fat, far from a mere aesthetic woe, hastens aging and dulls metabolism. It elevates risks for diabetes, heart disease, and other chronic ailments. People often lose muscle and gain body fat as they age, even when their body weight remains the same, Wang observed. The relentless production of fat cells by committed preadipocytes, age-specific, especially around the abdomen, fuels this insidious cycle. What lies ahead? Scientists aim to track CPA cells in animal models and humans, seeking ways to neutralize or block them. Could we halt age-related fat gain at its source? The prospect tantalizes. Could we halt age-related fat gain at its source? The prospect tantalizes. By unraveling the role of committed preadipocytes, age-specific in metabolic disorders, researchers edge closer to therapies that could redefine healthy aging. This pioneering work, led by first authors Guan Wang, PhD, of City of Hope, and Gaoyan Unele Li, PhD, of UCLA, alongside co-corresponding authors Wang and Xia Yang, PhD, of UCLA, sets a bold course. Their collaboration harnessed single-cell RNA sequencing to compare APC gene activity, revealing how middle-aged adipocyte progenitor cells roar to life while their youthful counterparts slumber. Such insights could reshape our battle against obesity. To support our channel's growth and ensure broader awareness, kindly hit the like, share, and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.